Okay, so today we are going to be talking about purification of immunoglobulin from rabbit serum in order to practice some affinity chromatography techniques. So let's first talk about some immunology terms. The first one is, of course, antibody. Antibody is a protein that's produced by our immune system, and it's going to bind to some type of an antigen that it's being made against. An antigen is the molecule that is getting recognized by the antibody that we are utilizing. Now, any molecule that the antibody is being made to is usually a pretty large molecule as far as the stuff is concerned. It's going to contain many parts and will have lots of other amino acids or lots of other parts as well. So the exact portion of that antigen that molecule that the antibody is going to bind to is what we call an epitope. So antibodies can be made against different epitopes on the same molecule. All of them would recognize the same protein or the same molecule, and they will all bind to the same molecule and be able to isolate it or recognize it, but they will be binding to different areas on there. So the same antigen can have many epitopes on them, each one binding to a different antibody. Now, there are two different ways that we can create antibodies, and they are polyclonal and monoclonal. In a polyclonal antibody mixture, we have many different antibodies that are made to the same antigen. Each one is, going to be recognizing a slightly different area on your protein, so you will have different parts of your antigen getting recognized by these different ones. This gives you more coverage in some ways, so you can have more molecules binding to it because of all these different antibodies that are made to the same antigen at the end of the day. In a monoclonal antibody, there is a very clean, purified antibody against a specific epitope. And that can be extremely helpful if we were looking for a very particular part of our protein. Maybe we are looking at the active part of the enzyme, that little active pocket, and we use that as an epitope to create our antibody. Or maybe we are looking at like the phosphorylated state of our protein, so we are specifically using that phospho antibody protein as our antigen to create the antibody against. So monoclonal antibodies have a very a lot of usefulness in the research community. But those are the two different ways that we can make antibodies, polyclonal and monoclonal. Polyclonal obviously allows you to get more coverage, so more proteins separated out. Now, most antibodies have some parts that are going to be constant all the time, and then obviously that part that's going to bind to the antigen is going to be a variable part. But antibodies have a very specific structure that's going to be constant. They are made as heterodimers, so they are made of 4 polypeptide chains. 2 of them are what we call heavy chains, but they're larger, and 2 of them are light chains, and they're smaller. The heavy chains are about 55 kilodalton, and your light chains are 22 kilodaltons. And they come together in the quaternary structure to create this classic Y-shaped pattern. The light chains are bound to your heavy chains with disulfide bonds, Sol linkages, hydrogen bonding, all those things that we've talked about before when we talk about protein structure and how they make the protein folding. So the light chains are linked with your heavy chains with these disulfide linkages, and then there are all within them as well. There are going to be many places where you will have your typical hydrogen bonding, hydrophobic interactions, all that good stuff. The 2 heavy chains are again bound together the same way. They have a lot more disulfide bridges between them to make a stronger bond so that they can be together in that classic Y-shaped structure. Now, when you look at a typical structure, you have your heavy chains on the inside, you have your light chains on the outside. The top part, the amino terminal, is where typically you have your antigen binding sites. That's where that's going to happen. This top region of both the heavy and the light chain is what we call the variable region. That's what's going to change depending on what epitope it's going to be binding to, what molecule it's going to be against. And then your constant regions, the rest of your protein in the antibody, all these other parts are your constant region. And they remain the same no matter what it is. So the variable region are your both present on your heavy and the light chains. They are the top 110 amino acids or so in the amino terminal. And they are going to be your complementary determining region or CDR regions that are going to contain specific amino acids. Those amino acids change based on what antigen they are going to be binding to, so or what epitope they're going to be recognizing on those antigens. Those amino acid sequences are going to provide the specificity on how strongly they will bind to a particular antigen on there. So these are what we call the antigen binding sites. And then your variable constant regions, the constant regions are going to be the same amino acids no matter what. So here you can look at different ways that these antigens can be binding to your antibodies. They're not binding typically just on one little tiny area, although sometimes you can have that. Usually the antigen-antibody interactions happen over a broad surface, so over a larger surface area on the outside of that structure, on that protein that you are binding to by your antibody. So all along your, essentially your heavy and the light chains region, all of those are going to be binding to a portion of your antigen, and that area that they bind to is what we are going to call our epitope. How do we make these antibodies? Well, antibodies are made in living organisms, and they're found in serum. So you can use essentially any living thing that can do that to create that. It's part of our immune system. They're part of our immune response. And that's part of our ability to differentiate from self versus non-self. So our blood is obviously composed of all kinds of different things. It has the plasma fluid that all the other cells are in, and then it has your red blood cells, your erythrocytes, it has all the immune cells, the leukocytes, and then your platelets. So your plasma contains, all these molecules, including the macromolecules used for clotting, which are your platelets and other molecules associated with it. Now the serum is the fluid phase that remains once you clot the blood and remove all that stuff from there. The serum, so if you take a vial of blood and you let it clot, you remove that portion, so it's going to remove all the platelets, all the erythrocytes, a lot of that stuff. What's going to be left behind are going to be your albumin and alpha, beta, and gamma globulin fractions. The antibodies are found in your gamma globulin fraction. Those are your immunoglobulins. Those are the ones that are going to be producing your antibodies. So here you can see that in response. In a diagram, you have your immune system that is getting run by a lot of different systems, including your lymph nodes, your spleen. And it contains your bone marrow, contains all the different, the stem cells that are going to create all these different specialized immune cells. And that's, again, part of your innate and adaptive immunity response. It's your adaptive immunity response that's going to give rise to the antibody. Okay, so as we talked about, that's how we produce our immune response as well. And these antibodies are then going to be protecting us as well. Also, if you have an autoimmune disorder, these same antibodies can come back to haunt us and cause damage to our own systems if they do not recognize self versus non-self accurately. So how do we produce antibodies in a research or in a commercial setting is by using, again, other living systems as our system of input, as our factories, so to speak. Commonly we use mice, rabbits, or pigs, sometimes sheep, and even humans to create these antibodies. So we use any of those systems as our system for producing, as our factories essentially to produce our antibodies. They are produced by the B cells within the immune system. So what we do in this case is we are going to inject the antigen. It could be a part of a protein that we want to use as an epitope, or it could be the full protein. If we just want a polyclonal antibody, for example, and we inject this antigen into the, into whatever system we're going to use, whatever animal we're going to use. So in this case, rabbits. This antigen would then be recognized as non-self by our B cells, and they are going to bind to that antigen. Once these B cells bind to that particular area of, bind to a part of that antigen, of that fragment or that full protein that has been given, they are going to do two things. They are going to produce some plasma B cells that can recognize it, but they're also going to be producing some B cells that are called memory B cells. And these memory B cells are going to create binding sites for all those places that it bound to. So by doing this, all these, little antibodies that are getting made, that are getting expressed on the cell surface of these memory B cells are essentially your antibodies. If you were to just take all of them off, you're going to get a mixture of antibodies made from different parts of those different epitopes binding to that original B cell. So they are going to be what we call polyclonal antibodies. And then we could also separate them out and get a monoclonal antibody against the specific portion as well. To isolate these antibodies, what we do is we take the secreted antibodies in the plasma by collecting the blood, allowing it to clot, and taking the serum from those rabbits. And that's going to contain all those monoclonal antibodies against that antigen that we have put into that particular animal. So anytime we are going to be, again, injecting an animal with our antigen, you're going to have several different B cells binding to it, producing a mixture, and that's going to be creating our polyclonal antibodies. That's usually the easiest ones to get, and we can use them. You can also create monoclonal antibodies by specifically altering this method. So once we have injected our antigen into the animal, we can isolate some spleen cells, and we can look at what different mixtures are present there. We can also isolate, obviously, our plasma cells and look at that as well. In this case, typically what we do is we fuse these cells, the plasma cells, containing those mixtures of antibodies, with myeloma cells, which is another type of cancer, actually, a blood cancer. But these myeloma cells, because they are cancer cells, can grow in culture a lot longer, they can be utilized, and we create what we call hybridomas. In a hybridoma, it's a fusion of those plasma cells that we isolated from the spleen and then your myeloma cells. Now, each one of them is going to have different types of antigens getting expressed depending on which cell was bound to it. The spleen cells that we get will have, each one of them will only be expressing one of those many antigens that you were initially looking at. So these hybridomas can then be separated out so that you have about approximately one cell per well or per dish and grown. And those clones are then going to be giving you what we call monoclonal antibodies, where each antibody fragment that we get, or each antibody fraction that we get, is going to be only against one specific epitope. And so this is how you would get a very clean antibody against a very particular epitope of choice. And then we can utilize it to study the function or to isolate that particular area of our protein. So the biggest difference is that your monoclonal antibody is going to be specific to only one epitope, while a polyclonal antibody is going to be a mixture of antibodies where a lot of different epitopes from the same antigen, same protein, are being expressed. So here, again, it's showing you that process where you have your antigen in your mouse. It's going to make that antibody. The B cell lymphocytes die after a few days in culture, but if you create these hybridomas, you're going to get indefinite culture you can grow them in different wells and you can isolate different monoclonal antibodies. We usually use selective medium in these so that only the hybridoma cells survive and none of the ones that are just the plasma cells or just the myeloma cells survive, based on the selection criteria that we use. And then we usually take, because obviously now they're in culture, they're just going to secrete their antibodies, their proteins into the media around them. We can just look at the media that is around the cells and look at which one is expressing what type of antibodies, so that we can get just the one that we want from there. So other things to remember when we are looking at antibodies, there are different types of antibody isotypes that are produced even in this category of antibody production. So there are IgGs. These are the largest population of antibodies. These are soluble proteins. They're going to be the easiest and the biggest fraction to find. Then you have IgNs that are going to be B-cell receptor membrane bound, and so some of it is going to be soluble, but a lot of it is going to be bound to those B cells and allow for the recognition and binding of that antigen. You also have IgDs, which are also membrane bound, and we don't exactly know what they are utilized for, but it is thought to be a co-receptor to your IgMs in the B cells. And then you have IgAs that are secreted as a soluble protein. And finally, you have IgEs, which are responsible for the allergic reactions that we get. And most of the ones that we are going to be looking at would be the IgGs, because that's, again, the largest population. These are all found in different parts of the body as well, and this just tells you where you might be able to see them. IgAs, because they're secreted, can be found in a lot of the secretory systems, so they can be found in mucus and saliva, tears, even breast milks, and they could be part of that immunity given to the baby because of it. IgDs, again, are going to be part of your B cells, membranes to be part of that receptors. IgEs are going to be responsible for allergic reactions. So, how do we isolate our Ig isotypes, the various ones? Well, there are certain bacterial proteins that are known to have high affinity for Ig isotypes. There are three different ones that we are going to be looking at. Protein A, protein G, and protein L. Protein A has very strong affinity to subclasses of IgG proteins from rabbit, pig, dog, and cat. Like I said, a lot of our antibodies are going to be prone in rabbit, pig, mice, so that's a very good one to use if you have one of the ones that has strong affinity to. So, for example, rabbit. Protein G binds to subclasses of mouse and human, again, two common antibody producers. And protein L is going to bind to subclasses of the kappa light chain, so it's not going to be as specific. It will just bind any isotype as long as the specific kappa light chain is present that it binds to. So in our case, because we are going to be isolating rabbit IgGs, we would want to use protein A as part of our resin to isolate. So again, to remind you, we're going to be using a technique called protein affinity chromatography. It's going to take advantage of this high affinity of certain molecules to bind to a specific chemical or group. In our case, we are going to be isolating rabbit IgGs, and it has a high affinity with this protein A from the bacteria. So we are going to bind, that's going to be our chemical group. The matrix within the column that we have is going to have protein A bound to that. When we add our protein mixture, our rabbit IgG, our rabbit proteins, serums, all the IgGs within them are going to bind to the matrix because of its high affinity to protein A, and it's going to get all bound within it. We are then going to wash with different buffers to remove any unbound substances, so all the other serum proteins that are not IgGs. And then finally, we are going to use a high concentration of a soluble form of our protein A buffer so that we can elute away our IgGs. And that's going to then give us our purified rabbit IgG. So again, the IgG molecules in the serum have high affinity both to protein A and G within the matrix. In our case, because we are looking at rabbit IgG, so protein A is the one that it's going to be binding to. We are going to remove all those other Ig molecules with washing with a mild buffer. And then to elute our IgGs, we are going to alter the pH so that our association is disrupted and you can take out your IgG fraction, the purified IgG fraction. We are going to check the presence of our protein in the eluate by looking at A280, which is going to look at amino acid presence in our sample. And we are going to neutralize our pH to prevent our protein from denaturing so we can look at it further. So that's our lab for today, using affinity chromatography with protein AG column to isolate rabbit IgG from the serum.